All right, hello, dear 12. So, this is the final part of respiration, and that is gas exchange and transport in the blood. So we've talked about the anatomy of the respiratory system. You got the nares, the nasal passages, the mouth. Then at the back of the throat is the pharynx. You start to move down, you have the epiglottis, the little flap that closes over the trachea, and you got the trachea. Right then there is the larynx with the vocal cords. Go down the trachea, you've got the bronchi. The bronchi then branch off into bronchioles, and then the bronchioles end with alveoli, those little millions and millions of those little sacs where the gas exchange happens. And that's what we're going to be talking about today, is that gas exchange. The other thing we talked about is the mechanics of breathing. So when those alveoli stretch, they send a signal to the medulla oblongata to stop inhaling. The medulla oblongata cuts off the signal that it's been sending to the intercostal muscles, which have been pulling the rib cage apart, and the diaphragm at the bottom here. The diaphragm has been contracting, and it relaxes. It makes the thoracic cavity smaller. The gravity brings the rib cage down. The thoracic cavity gets smaller. Air is pushed out of the lungs. CO2 starts to build up in the blood. Your medulla oblongata detects the buildup of CO2, and it initiates an inhalation. Inhalation is active because it's a signal being sent from the medulla oblongata to contract muscles, and exhalation is passive. There is no signal being sent, and the muscles are turning off, are relaxing, going into their natural state. You can't breathe in when you're dead, but you can breathe out when you're dead. Right, so back to this inhalation. You breathe in, and what happens is air enters, it makes it to those alveoli, but the air hasn't made it into your body yet. It's not actually in your body yet. We don't consider it in the body. What has to happen first is what's called external respiration. Can I flip over this thing? Oh no, something's weird. Oh, it's the focus thing is weird. Oh, hang on. There we go. Okay. So external respiration. So if you imagine in this this diagram here, this is the inside of that sac of the alveoli. This is inside your lung, one of those millions and millions of sacs. And ignore this bit for a second. We're just looking here. So you've just breathed in, so there's lots of oxygen in here, lots of O2. Not a lot of O2 in here. So there is a concentration gradient. Remember, we learned about concentration gradients. And for gases, those are called pressure gradients. But it's the same thing as a concentration gradient. So there's more oxygen here, less oxygen here. What's happening is in that alveoli where all the oxygens are, they're bumping into each other. And they're basically, if there's a place where there's no oxygens to bump into, they're going to get bumped into there. And there happens to be the inside of the capillary. There is very little through here. There's just actually two layers of cells. There's one layer here, and there is one layer. There's one layer that's the wall of the alveoli, and there's one layer that's the wall of the capillary. And there's moisture here, so there's water which helps the oxygen move across. And obviously, there's lots of in the membranes and stuff here, there's ways for the oxygen to get through this whole area. That's its modus operandi. So more oxygen here, less oxygen here. So what you're going to get is you're going to get oxygen diffusing into here. And then as soon as it diffuses into the plasma, there is a protein, an enzyme, on the cell membrane of red blood cells called hemoglobin. The shorthand for it is this HB and little b. And what happens to it is it becomes oxyhemoglobin. it is to bind oxygen. And hemoglobin is it's got quaternary structure. Remember that from our enzyme unit. So it's actually 4 peptide chains, and they have what's called a heme, which is and it has an iron atom in it, and the iron atom rusts basically. And what happens is an oxygen is attached to that iron. So each hemoglobin enzyme carries 4 oxygen atoms, and it likes to bind. So what happens is now you've got oxyhemoglobin, and if you notice, when it binds to the membrane of the hemoglobin here, it's no longer in the plasma, which means that pressure gradient is maintained. It never can equalize. So whatever oxygen is in here constantly wants to move into here, because as soon as it makes it into here, it binds to a red blood cell, which then actually carries it away. So the pressure gradient is always maintained. Okay, this is the pulmonary circuit. Remember the pulmonary circuit from our circulation thing? All of these things combine. And so then that red blood cell now that is soaked in oxygen, there'll be billions and billions and billions of hemoglobins all over this red blood cell and carrying billions and billions and billions and billions of oxygen atoms. Apparently 96% of all the available sites to carry hemoglobin are saturated with oxygen. To carry oxygen are saturated with oxygen at this point. So oxygen is carried through the blood primarily, like almost 100%, on hemoglobin. Of it, I would say maybe a tiny little bit will be dissolved in the blood, but I would say that but tiny amount, trace amount. So, okay, so then it travels through the pulmonary circuit. It enters the left atrium of the heart, remember from our circulation unit, then enters the left ventricle, and then is squirted out. Left ventricle is the big one, the powerhouse of the heart. It shoots all those oxygen-saturated red blood cells out into the systemic circuit, so out into the tissues. So here we have whatever cells in the body. These could be brain cells, muscle cells, smooth muscle of your digestive system, cells inside your mouth excreting enzymes, whatever cells in your body that are doing stuff in the tissues. There is a capillary near them, that very close to them, there are billions and billions of capillaries that will have a red blood cell full of oxygen coming near them. Now, notice something. Over here, you'll notice a temperature and a pH. Here in the tissues, it's 38 degrees centigrade, and the pH is 7.35. If we look over here in the alveoli, in the capillaries of the alveoli, so in the lungs, it's a little bit cooler. It's 37 degrees, and the pH is 7.38. It's a little bit higher, a tiny bit higher, 0.03 higher pH, which means it's a little bit more basic. Now, remember from all the way back in our enzyme unit, enzymes and proteins have an ideal pH that they work in and an ideal temperature that they work in. As the temperature changes and the pH changes, the shape of the protein or the shape of the enzyme, that tertiary shape, will change. And this is true for hemoglobin. Hemoglobin is very sensitive to pH and very sensitive to temperature. And what happens when the temperature is a little bit warmer, 38 degrees centigrade, and the pH is 7.35, a little bit more acidic than it was in the capillaries of the alveoli, hemoglobin does not want, its shape changes and it loses some of its affinity for those oxygen atoms. Doesn't want to hang on to that oxygen so much. And so what ends up happening, the capillaries, are only one cell thick. The red blood cells have to squeeze through the capillary. And here, remember, they're under pressure. There's also huge amounts of pressure. Remember from our circulation unit, there's the pressure of the heart beating is actually pushing water out of the capillary. And as these are bouncing along, the oxygens get shaken off. And if you imagine little oxygen bubbles, they won't look like that, but little oxygen atoms getting shaken off, and the water, the plasma, is getting pushed out of the capillary, and along with it come those free oxygens now that hemoglobin didn't want to hang on to so well because the conditions were different. And now those oxygens are in the extracellular fluid, so they're out of the capillary, they're in the space between cells. And the cells are like, now are like, yay, our oxygen delivery has come. We can continue doing cellular respiration and metabolism and all that stuff. And they immediately suck up that oxygen to do cellular respiration. And so the pressure gradient, again, is maintained because those cells are eating up that oxygen. So whatever oxygen is in here, there's a greater concentration of oxygen that then goes into the extracellular fluid and then gets sucked into the cells. So whatever oxygen's in the extracellular fluid, there's a pressure gradient, so the oxygen wants to go in there. And the cells are grabbing it, are actively grabbing it. So there's always a pressure gradient for oxygen to get out. Now, the pH is 7.35, the temperature is 38.7, and now hemoglobin has changed shape, and it hasn't become useless. It hasn't become broken or anything like that. In fact, what's happened, it now has an affinity, it's changed shape. Now it has more of an affinity for carbon dioxide. Not for oxygen now it wants to hang on to carbon dioxide. How convenient, because the product of cellular respiration, that oxygen that's gone in, the stuff in the mitochondria happens where you're making ATP. The product of that is carbon dioxide, and carbon dioxide is toxic. So we want to get rid of the carbon dioxide. So what happens is two things happen. Here it gets a little bit crazy, and you can see it's a little bit small here. I wonder if I can I zoom in on that? Is there a way to zoom in? Can I zoom? View, zoom. There doesn't appear to be a way for me to zoom in here, like on the other computer. Oh, now I feel like an old man. Anyways, okay, but that's okay. So let's just follow. So there's a lot of carbon dioxide here now. These cells have been metabolizing. They're producing carbon dioxide. And actually the reason this carbon dioxide turns into carbonic acid, which is what lowers the pH slightly. So the carbon dioxide is lowering the pH, which makes hemoglobin, the HB, have more affinity for carbon dioxide. But there's another enzyme in the membrane of red blood cells called carbonic anhydrase. And what carbonic anhydrase does, so the product of cellular respiration is water and carbon dioxide. So it takes the water and it takes the carbon dioxide, and even though they would do this naturally, but it speeds it up, is it turns the carbon dioxide into the acid. It turns it into acid, which you'd think is bad, but it's actually good. Because what happens is that acid immediately, because it's in water, it dissociates. And it dissociates into bicarbonate. Do I have that formula here? It dissociates into bicarbonate and those free hydrogen ions, which are bad. Remember, the hydrogen ions are bad. They're the positive hydrogen ions. But there is a rescue for this, and that is hemoglobin. So this is all happening on the membrane of the red blood cells. I know this is crazy. This is all happening on the membrane of the red blood cells. So on the membrane, carbonic anhydrase is taking water, it's taking the carbon dioxide, and it's turning it into acid, which then quickly dissociates into these hydrogen ions and bicarbonate ions. And then immediately, without hesitation, hemoglobin in these conditions really, really likes those hydrogen ions. So before these hydrogen ions can do any damage, they attach to that hemoglobin, which becomes what's called a reduced hemoglobin. And it's HHB, reduced hemoglobin. So those hydrogen ions can't cause any damage. The bicarbonate ions, those are good. Those actually, they buffer in blood plasma. They're a buffer. And what a buffer does is it maintains pH. So what they will do is they're like a weak base. So what will happen is any hydrogens that are in, any leftover hydrogens will actually associate with the bicarbonate. And they buffer, they maintain the pH. So now this is what happens to the majority of CO2. The majority of CO2, the water and the CO2 get turned into carbonic acid. And let's see if I've got that formula here. There it is. So this is what's happening. CO2 plus water turn into this carbonic acid, H2CO3, and then which immediately dissociates to hydrogen and bicarbonate, and then immediately that hydrogen joins with the hemoglobin, and it becomes reduced hemoglobin. So that's the major way. That's the number one way that carbon dioxide is carried in the blood, okay? Now, also, some of the carbon dioxide, hemoglobin, it doesn't do this. It just attaches to the hemoglobin. Just joins with hemoglobin. Hemoglobin now, because of that pH, because of that, is also attracted to CO2. So, and that is called carbaminohemoglobin. So you've got reduced hemoglobin, carbaminohemoglobin. Okay, that's all that enzyme, that's all that protein on the surface of the red blood cells. And then some of the CO2, a small amount, just travels in the blood as CO2, just dissolved in the blood. So that is all the ways that now CO2 gets carried. It's three ways. Oxygen just gets carried on the hemoglobin. Let's just say it just gets carried on the hemoglobin. So oxygen, really easy. But carbon dioxide, we've got three ways that it travels through the blood. The major way is that carbonic anhydrase turns it into bicarbonate and hydrogen ion, basically. Carbonic anhydrase turns it into the acid, then the acid dissociates into bicarbonate and those hydrogen ions, which then immediately merge with hemoglobin. So it's carried as reduced hemoglobin and bicarbonate. Then a smaller amount is carried as carbaminohemoglobin, the carbon dioxide immediately merges with the hemoglobin, and then some of it is dissolved in the blood. Now, probably one of the reason I would say is because CO2 is toxic, we've got three ways to carry it out of the blood out to make sure we carry it out. And if you notice, two of those ways, it's bound up, it's locked up it's changed and locked up in proteins, and in fact, it's changed into a buffer so that the effects of acid are reduced. So then, let's continue on. What this is called in the tissues, what we just talked about, is called internal respiration. So that's internal respiration, and external respiration is what happens in the alveoli. Internal respiration happens in the tissues, external respiration happens in the alveoli. Probably a question on a test, will be something about internal respiration. Where's internal respiration? And if it's a multiple choice, students often put in the lungs. They'll think because it's internal in the lungs. Don't be those students. Know that internal respiration is in the tissues. Okay, moving on. So then our red blood cells now are on their surface are carrying reduced hemoglobin, which has got the hydrogen ions, HHB hemoglobin, and carbaminohemoglobin, HbCO2. And there's also in the plasma now, there is carbon dioxide, just bubbles of carbon dioxide dissolved in the plasma. Sure, let's say that. And there's the buffer bicarbonate. So there's going to be a lot of buffer bicarbonate, a lot of HHB, some HCO2, and very little CO2. And they travel. Now they're in the veins here, right? These are the veins. This is deoxygenated blood traveling in the veins towards the heart. They enter the right atrium of the heart, then the right ventricle into the pulmonary circuit, and now they are back into the lungs And what's happening here now is the conditions change. It's cooler here because this is the outside world, and it's more basic here. And so what happens is hemoglobin changes shape again. The tertiary structure changes shape again. It doesn't want to hang on to this carbon dioxide anymore, so it lets it go into the plasma. At the same time here, carbonic anhydrase, this enzyme, also changes shape in these conditions, and now what it starts doing is the opposite of what it was doing before. It starts taking hydrogen ions and bicarbonate and merging them together. No, wait. That's right, sure. And then splitting them off into CO2 and water. And some of the water you will actually breathe out. Your air is moist. When it's really cold, when you breathe out, you can see that's not smoke or something like that. That's the product of cellular respiration condensing in the cold air and turning into water vapor that you can see. And now the plasma here, because hemoglobin has let go of the CO2, hemoglobin has let go of that hydrogen, carbonic anhydrase has turned that hydrogen and the bicarbonate into more CO2. Now there's lots of CO2 inside the plasma in this capillary. There's no CO2 in here, so what we get is we have a partial pressure, so it diffuses out into here. Out it goes. Meanwhile, of course, now all of these hemoglobins are free to take up oxygen as they move through, and you breathe out that CO2 and the water vapor in external respiration. And that is gas exchange and transport in the blood. So there you go. In your mind, what is the story of an oxygen atom would be a good thing to go through, right? So an oxygen atom enters here and merges with hemoglobin. It goes on a real adventure, a real hero's journey. Merges with hemoglobin. It travels through the left atrium, the left ventricle, through the arteries. It travels to the tissues. Then it's let go hemoglobin. It's the love affair with hemoglobin ends here. It's a very short love affair, and out it goes into the external tissues. And then it's embraced. Its breakup. What's that called? When the rebound is these cells, but the cells, they're a bad rebound. They just end up using the O2. In cellular respiration in their mitochondria, and then that oxygen gets basically merged to a carbon or a hydrogen. It becomes water, and then it's let go. The love affair, it's changed. It gets changed by its love affair here, and then it travels here maybe as bicarbonate, maybe as just CO2 attached to a hemoglobin, maybe as CO2 just dissolved in the blood. Out it goes in the veins, then in the pulmonary arteries, and out back into the world, out into the air to do whatever it's going to do out there. So review that, have a look at, and that basically concludes respiration. You can look on your own at respiratory diseases. There you go. Okay.